Okay, so that's point mutations. They create new alleles, right? Everyone's okay, comfortable with the terminology of genes versus alleles. You better be, because you'll need to be comfortable with it. Now, new genes. How do you get new genes? You go to the Gap and you buy new genes. All right. The major way that you get new genes is through what's called gene duplication. And if you have unequal crossover at meiosis, then you can have one of the daughter chromatids have an extra copy of a section of DNA and one completely missing that section of DNA, right? So here's this diagram of crossover of homologous chromosomes, right? And what you see, or I hope that you see, is that in this section where there's crossover, you're not getting exactly correct alignment and swapping over. It's unequal between these chromatids, so that what you get as a result, or you can get as a result, is of these four daughter chromatids, you have one that is missing the C gene. What's likely to happen to the zygote that's produced by that gamete? It's probably dead, right? If the C gene does anything useful, right? Probably never develops or miscarries or something like that. What about zygotes that are produced by, I'll switch to blue here, this one that has an extra copy of the C gene? Is it going to be dead? Why not? It needs whatever gene product C codes for, and it's got it, right? It might be overexpressed. There could be different sorts of other kinds of problems. But this unequal crossover has now duplicated the C gene in a way that could allow it to increase in frequency in the population. And now the population of organisms can have two copies of these genes. So why is that important? Those copies can diverge in function. That's what you're going to say, right? Okay. So here's a quick little example of the globin gene. So gene duplication has been incredibly important in providing raw material for adaptive change. Your hemoglobin molecule has 4 subunits. In adult humans, 2 are from this alpha cluster on chromosome 16, 2 are from the beta cluster on chromosome 11. I know you don't really care. When you were a fetus, there were different variants that were used to make up your hemoglobin molecule. And they have different oxygen affinity. So the fetus is developing within the mother. It's getting oxygen from the mother's blood supply via the placenta, right? If fetal hemoglobin had the exact same affinity for oxygen as maternal hemoglobin did, what would be the gradient of oxygen supplied the fetus? It would be less than if the fetal hemoglobin has higher affinity for oxygen than the adult. The point is that all of these different hemoglobin molecules that are put together, these subunits are put together, are encoded by copies of the same gene that have diverged in function. So this is obviously an adaptive kind of divergence in what the oxygen affinity of these molecules, but the duplication of that gene is what allowed the 2 copies to assume different functions, because one copy can retain the original function, which makes the other copy just extra. So it could just degrade, or it could diverge to have some kind of secondary related kind of function. And there's lots and lots and lots of